All right, so back to EBE. I think the last thing we were talking about was speciation. And we had finished discussing allopatric speciation, the idea of dividing a population into two subpopulations, and then each of those two subpopulations is free to evolve separately. The lack of migration between these two subpopulations allows for genetic differentiation to occur, and as this differentiation begins to increase, eventually these two populations are no longer to produce offspring together. Okay? The second model that we have for speciation is a sympatric model. Unlike allopatric, sympatric does not involve separating the population into two subpopulations. The speciation process is going to occur with everybody in the same place, and it's going to occur instantly. With allopatric, the separation is not the speciation event. Separating the subpopulations from each other allows for the differentiation, which sets up the long-term process of speciation. However, sympatric is going to occur instantly. So I have a couple of examples to do with you. Let me go ahead and start this first one. So what we have in the United States on the East Coast, up in New England and into New York, is seagrass here that lives in coastal marshes called Spartina alterniflora. Okay, it's native to the United States on the East Coast. It's also invasive on the West Coast, but it's native to the East Coast of the United States. Then we have the Atlantic Ocean here, and then in Great Britain, we have another closely related species called Spartina maritima. Okay, Spartina maritima. They're closely related species. However, in typical situations, they don't interbreed together. That's why they're separate species. I mean, you could make a decent argument maybe these species evolved back around the time of Pangea, and then the separation of the continents, the increasing distance between North America and Great Britain, maybe this allowed these two species to separate from what was one original Spartina species. But in any event, this is kind of the way things are now. If we go back to the Age of Discovery, one of the things that was going on in the Age of Discovery was the establishment of these colonies. One of those colonies was in the United States, right? The original 13 colonies. And there were trade routes that were set up between Great Britain and the colonies that Great Britain controlled the majority of. So if you kind of think about what goods and services are available in these two parts of the world, Britain has a heavy industrial foundation at this time. The colonies do not. So one of the things that they lack are tools and anything made out of iron, because they don't have any foundries to manufacture this stuff, right? The raw materials that they would use to blacksmith into horseshoes or plows or rakes or whatever, they need that raw material from Britain. Britain, on the other hand, needs food. There are a lot of agricultural crops available in the colonies. You may be familiar with tobacco, but originally the big trade good here, the agricultural crop in the colonies, was sarsaparilla. is the species name, but it's called wild sarsaparilla. The rhizome of this plant has a root beer flavor to it. So you may have had sarsaparilla soda, that's from sarsaparilla. I always kind of imagine back in the 1600s, 1700s that water must have just tasted terrible, and that's why something like a root beer flavoring would have been so valuable back then. But in any event, that was the biggest cash crop in the colonies before we colonized further south into places like Virginia and North Carolina. Right, in any event, you have this rich agricultural base in the colonies and an industrial base in Great Britain. So what you would do is take your ship here in Great Britain and you would load it up with iron. So you imagine you've loaded all this iron onto the boat. That makes the boat sink pretty low into the water. And this is actually stable for ships. That way they don't blow over when the wind blows. You want it riding low in the water. So the trade route, you go to the colonies, and then you unload all of this iron, and now you're riding high in the water. Well, you don't want that. You pick up your agricultural crops. Well, they don't weigh very much, so you're still riding pretty high in the water. So what you do is you take on what's called ballast water. And the ballast water allows the ship to sink lower in the water. You actually take up the water into these ballast compartments, and that helps pull the ship down so that it's more stable. Then you run back to Great Britain, you offload the agricultural goods, and then you're going to try to unload the iron. But you can't unload the iron until you get rid of the ballast water. So what you would do is allow this ballast water to be emptied out in Great Britain so that you could load the iron on. Well, some of the seeds of Spartina alterniflora got picked up by the ballast water. So the seeds were in the ballast water in the ballast compartments. The seeds get dumped into the North Atlantic in Great Britain, and some of these seeds established here, and now you've got Spartina maritima, the native species, but you also have Spartina alterniflora, which would now be an introduced species to Great Britain. Okay? Now, they're two separate species, so the idea is they shouldn't produce offspring together. If they produced offspring together, we wouldn't call them two separate species. But in plants, you can get something unusual occur that isn't as easy to do in most of the animals. All right, so in order to understand this, we have to go back to meiosis. And I don't want to belabor meiosis. I know meiosis is torture. The only thing I can tell you is that a good understanding of meiosis is critical, and so much so that you're going to get it again in your genetics course, whether it's genetics or human genetics. But let's take a simple system here. You draw my chromosomes in a slightly different orientation. Let's draw them like this. So here is a tetrad. Here's another tetrad, and whoops. It's been a while since I've done genetics. Here's a tetrad, and right, so there's a tetrad, and there's a tetrad. All right, that would be metaphase 1. And then after the first round of meiosis, we would end up in prophase 2 and running through the second round of divisions in meiosis. The way I've set things up here, this is what metaphase 2 would look like, given our original configuration in metaphase 1. So this is metaphase 2. Okay? And then based on this configuration in metaphase 2, you should end up with 4 cells. Oops. I'm going to draw them with the chromosomes, even though by the end of meiosis it should be back to chromatin. So meiosis starts with 1 cell, gives you 4 cells. I've just shown metaphase 1, 2, and the final configuration. So the idea is that if I were to label these chromatids, those would be those chromatids. These 2 chromatids are here and here. These 2 are here and here. And these 2 are here and here. So this is the way that meiosis is supposed to progress. But let's say that there was a goof up in meiosis. Mistakes were made, right? And one of those goof ups that can happen is what's referred to as a non-disjunction. And a non-disjunction is where the chromosomes don't pull apart properly. So instead of having the tetrads separate here, one of the things I could do is mess up those spindle fibers so that they don't work properly. Because these spindle fibers don't work properly, I don't end up pulling these 2 chromosomes to the opposite side of the cell. Instead, I end up pulling all 4 of these to the top. Then when I get to metaphase 2, this cell has all 4 chromosomes and this cell didn't get any, which means when I finish out here, it's hard to draw chromosomes. The more of them I draw, the worse they get. But now what I've got are cells here that have 4 chromosomes, which is the same as the 4 chromosomes I started with. Instead of the 2 in each, I now have 4 in 2 and 0 in 1. Does that make sense? Okay. All right. Now, the other thing that can happen is a non-disjunction event in the second round of meiosis. So everything went fine here. Right? But there's a mistake here. So maybe these spindle fibers didn't separate properly. And now everything would be fine in these top 2 cells. But here they went up, and I would end up with 4 here, and I'd end up with none there. So this is not correct, and it should look a lot like a non-disjunction in this first round of meiosis, where I had 2 cells with 4 and 2 cells with none. Sometimes students ask me, I'm trying to kind of anticipate questions people ask in class, sometimes students ask me, well, why didn't you draw them like this in that final cell? Well, keep in mind that the connection between these 2 chromatids at the centromere is not very strong, right? It's made to pull apart when you put tension on it. So it doesn't last forever, and eventually these 2 things are going to drift apart from each other if they're not pulled apart from each other. Okay? All right, so in any event, it's this kind of a cell I want to talk about. Starting with 4 chromosomes here, I should end up with a gamete that looks like this. Now, these are plants, so really that would be a spore at the end of meiosis, right? And then we would go through mitosis to get to the gametophyte, and then mitosis to produce the gametes. All of that occurs in here, so let's just refer to this as a gamete because we're talking about fertilization eventually. So if I take this one and cross it with another one like this, that gets me back to the original 4 chromosomes. And if this cell wanted to go through meiosis, it would go through a DNA replication phase to make the chromosomes look like that. So this is kind of the typical way this is supposed to go. Now, the haploid number here would be 2, and here the diploid number, because we're still in metaphase 1, or the original cell that we started with here, this would be a diploid of 4. Well, by definition, N is the number of chromosomes within a gamete, okay, by definition. So here I have 2 chromosomes in the gamete, that's an N of 2. Okay? All right, now, let's say that there was a goof up in meiosis, and all the chromosomes went to one side, and I made instead a gamete that looks like this. Okay? And let's say that exactly the same time that I made this goof up, in a completely different plant, I made the same goof in meiosis and generated this. So I doubled the chromosome number here above what it's supposed to be by messing up meiosis. Chromosomes all go to one side, but it happens in 2 different plants at exactly the same time. Chances of that happening, very rare, but it's a finite probability, so it could happen. That means that my new cell here would have 4 plus 4, 5, 6, 7, 8 chromosomes. And how many chromosomes in the gamete? Well, this is an N of 4. This is a mess up in meiosis,